All right, good afternoon. I got this. It's all set. Thank you. I'm going to talk about microbes for energy generation and wastewater remediation. And I'm happy to be here, but I'm really not happy to have to speak after Tal because they use clean rooms and their structures are really well defined. And what we're trying to do is really deal with messier systems. And I'm going to take a big leap of faith here, and I'm going to start with the first slide and with some chemistry. All right? And what I want to introduce is the fact that we tend to think about molecular design in the sense of how do we maximize function in a simple structure, in a simple molecular structure, and something that operates generally and that addresses an issue of interest. So I'll give you some background about these molecules. This is an outline of the talk. Then it turns out that these molecules, for reasons that we can talk about, actually they spontaneously insert into membranes, either artificial or in microbes. I'll show you, it's very easy, you don't have to do much work. And then when that happens, actually when they insert in the membrane of a microorganism, then all of a sudden, the way that that microorganism can relate to an external surface, like an electrode, changes considerably. We find that it's possible to remove electrons from the microbe. It's easier to put electrons into the microbe and, for example, stimulate metabolism. And I'll give you some, I'll show you that. And then once you do that, then you can sort of incorporate those microbes into a device. These are bioelectrochemical devices, such as microbial fuel cells, wastewater remediation. And then all of you, what you find out is the way that you can actually treat the food that you give it and convert it into an electrical signal is improved considerably. So that's basically what I want you to remember, and I'll give you now the details of how we got to that point. And I was asked to provide some background, and I'd like to thank Nate there for digging up some pretty early work. And so the work by Galvani back in 1791, kind of cool. He first demonstrated that you can, I guess, chop off the legs from a frog, and then you can put a zinc and I guess the copper electrode, and they would twitch. That was kind of cool because that kind of indicated that somehow this electrical stimulation, at least in this particular case, induced the contraction of the muscle. More relevant, back in 1911, I guess the work by Potter at Durham actually showed in a very direct way that our ability to breathe requires an electron transport chain. Oxygen is the final electron receptor, and that electron transport chain allows you to take food, convert it into CO2. That's important because that's the critical component in how we can convert waste into electricity. Now, current applications, I fast forward until today, people are actually taking advantage of those processes. For example, Cambrian Innovations is using microbial fuel cells to take waste and actually more efficiently process the wastewater, and in doing so, they actually can generate hydrogen, which then reuse and minimize the energy cost of treatment of water. And then beautiful work by Lenny Tender at Office of Naval Research. They basically use, they just go to the seafloor, they dig a hole, they put an electrode there, they put another electrode on top of the seafloor, and there's a chemical gradient there, and they can use that power to generate a sensor that the Navy has up on the surface. So these are sort of real current applications of that process. So the materials that we work with are actually derived from these conjugated polymers. These are the same polymers as John mentioned that are used in organic solar cells, in transistors, in OLEDs, that type of technology, where we put, in our case, we can make them polyelectrolytes, so they have the charge distribution, like for example, there's lots of polyelectrolytes out there, like DNA. And actually, we studied this class of materials a lot 10 years ago because we could use the backbone as a light harvesting unit, and deliver those excitations to specific dyes when there is a specific molecular recognition event. I'm summarizing a lot here, but I just want to show you that that fundamental work actually led to the creation, the founding of Sirigen company. They developed further these types of ideas, and now these materials are used in the most efficient dyes for flow cytometry. So these are not obscure materials that are difficult to obtain, but they're accessible. All right, so these are what the molecules look like. Just keep that in mind. There's a aromatic, a conjugated segment, there's ionic groups. But the important thing is that they have the same dimensions and charge distribution as a membrane. So when you add this molecule to solution, boom, spontaneously inserts into a membrane. And we verified that, I mean, through optical characterization studies. This is with confocal microscopy. The fact that we see a brighter region along this direction is consistent with the fact that the molecules insert like this. They don't just randomly absorb. And this is more of a schematic figure of what this really looks like. Now, these are artificial systems. And now I want to move forward to what happens when you use a microbe. And I'm going to start with yeast. And the following slides just will show you basically the microscopy of yeast on one side and the emission from the same yeast on the other side. Here's the yeast. I think it might be a little bit too bright for this to be observable. But in about 5 minutes, I don't know if you can see this, we have labeled just every single yeast with this dye and thereby modified the membrane. Okay? And it's within 5 minutes. Very new. Here, let me show you that again because it's kind of cool. So here it is. There's the timer. This is the microscope, and here's the emission from those molecules all around in the membrane of the yeast. Here's a tomographic image from a mammalian cell. You can see that it's only in the surface of the cell. And this is another experiment where we kind of bleached the molecules here, and over time, you can see that they move within the membrane and they repopulate that region that was originally bleached out. All right, so this shows you that this process works. It's simple. It's just a question of adding the molecule. And then the question is, what about the bioelectrochemical properties of the microorganisms? And I need to sort of define and show you how a microbial fuel cell works, because that's the test bed that we use for measuring how much electricity we can remove from the bacteria. And so you take bacteria, you put it on an electrode, and you don't give it oxygen. Just starving for oxygen, but you give it food. You can get glucose, you can add wastewater, all sorts of different things. But you can see for this bacteria to take the glucose into CO2, it needs to find an electron sink, and that would be oxygen. And that oxygen is located in a different chamber. This is another electrochemical, the other half cell. So for this to be able to breathe, it actually has to dump an electron onto that electrode. And that electron goes around and reacts with the oxygen, creates water with a concomitant motion of a proton from one side to the other. So this is the way this works. Keep it anaerobic, give it food, give it an electron where it's going to breathe into. Goes around, reacts with oxygen, and you complete the circuit. And so with Jim Sumner at the Army Research Lab, we worked on these types of cells. And this is proportional to the current that's generated. This is the yeast microbial fuel cell without any modification in green. And in red is the same yeast that I showed you that had been labeled. So you can see there's a dramatic increase in the amount of current that's being generated. All of a sudden, that very simple step of modification of the membrane renders these yeast much more amenable to communicating with the electrode and sending the current over, which is kind of neat because we didn't do any genetic engineering. It's just a simple way of changing the way that the metabolic pathways relate to the external circuit. And this works for a variety of different microbes. Here's E. coli. E. coli is kind of nice. There's innumerable mutants associated with it. It's spontaneously labeled. And we can do the same experiment with the microbial fuel cell. and we take, so we would compare the performance of wild type, unmodified E. coli versus this type of E. coli. And you can see in black, this is without modification. As you add more and more of the molecule, you get greater and greater current. It's kind of neat, all right? But this doesn't tell you the whole story about the fuel cell, because the fuel cell, you need to really determine the power density, not the total current. And in black, we show you what is obtained with just normal E. coli, and this is what happens when you've modified the E. coli as shown here. So what this tells you is that you've considerably reduced the internal resistance of the device. Okay, somehow by changing this membrane property, we can dump electrons, or the bacteria can, or the E. coli can dump electrons to the substrate. So these are well-controlled systems. We picked yeast. Army was interested in the yeast because it's less susceptible to antimicrobials, and they were interested in powering unattended sensors. In this case, we used E. coli. What about if we take a microbial community that we don't fully pre-program? And this is where the wastewater microbial fuel cell comes in. Wastewater has a bunch of waste, carbon content, and some microbial population that might be different here relative to San Francisco, relative to Africa. Variety of different components. And what you do is you basically set up a same type of microbial fuel cell. You just take the wastewater, you cap one end. There's no oxygen here. One electrode, another electrode here. This is open to oxygen. And we're going to compare what happens when you just use the wastewater and versus adding a small amount of this molecule to the wastewater. This was collaboration with Steve Harvey at the Edgewood Chemical Biological Center, because this Army base was trying to figure out ways to deal with some of the wastewater that we're generating. Anyway, so let's see what happens. And so here's the current that's being generated by this microbial fuel cell, here in blue. Right? There's not much current. Right? And this is what happens when you've added that molecule, the oligoelectrolyte, to the wastewater. You see that after, it takes time, okay? This is not a ultra-fast spectroscopy, right? This is different. This is 2 weeks. But after 2 weeks, that microbial population that came with the wastewater, in the presence of that molecule, actually evolves into a situation where it becomes electrogenic, meaning it can actually deliver the current or the electron equivalent onto the electrode, and it creates an electricity. So this is sort of interesting, because in this case, we don't particularly know what we started with, and we don't know the biofilm composition here. We don't know the biofilm composition here or there, and this is, of course, the kind of things we really want to move forward in our research. But unambiguously, we've been able to create a microbial community that actually can deliver electrons to that circuit. And here's some STM. There was some very nice cross-sectional TEM in the previous talk. We need to show some. These are the electrodes from that wastewater microbial fuel cell, and you can see the microorganisms that have populated and somehow have evolved the ability to generate current. What about other biofilms? What other systems can we look at to test whether this is general or not? And I'm going to borrow this slide from NSF, which is very similar to the wastewater microbial fuel cell I just discussed. We add waste. In this case, they want to use some kind of agricultural waste into our anaerobic chamber. And then you're going to create a current. And you want to add a little, about 0.2 volts additional bias by using an external source. Could be, for example, a solar cell. And under those conditions, those protons that were migrating over to this side can actually be reduced and generate hydrogen. So instead of just simply collecting the current, we just convert it into a fuel, hydrogen. And actually, this device is very simple to use. Here is the anode. cathode. This is what? This is a graphite anode. This is a cathode. It's a simple vial. Here are the leads. And you just add wastewater. And you add a little potential between these two leads. And it's a little bit more complicated. The way that you do this, you actually take the wastewater and you grow a biofilm, whatever that came in, all right? And that's going to be located here. You can add the wastewater, and the process begins as I told you. There's no membrane here, so you're just relying on some kind of concentration gradient for the hydrogen. Come over, so the protons are created for hydrogen. But there are other problems that can happen. For example, the hydrogen can be recycled. And more importantly, you can actually start generating methane. If you just want to make methane, that's fine. But that kind of cuts down the purity of the product that this biofilm is generating. So you're trying to sort of figure out ways by which this selectivity can be improved. And let's see what happens. We're going to just grow a biofilm from wastewater. We're going to treat one with the oligoelectrolyte, one not. We don't see any difference in the amount of current that's being generated in this particular case. And then let's see what the products are that come out. And so here we have, this is the product, either hydrogen or methane, and these are different cycles. This is the untreated. This is just a plain wastewater, and you can see that you're generating essentially methane. In the case of the treated biofilm with our molecule, it starts generating more hydrogen. Second cycle, more hydrogen, more hydrogen. And eventually you can see that the product selectivity is totally different. Right? In the original system, you just generated methane. In this system, you start biasing the product distribution towards hydrogen, which in this particular situation was the desirable product. Which is kind of neat. You know, biofilms, they're tough to control. We don't know how they communicate. They're very important in, for example, antimicrobial resistance. But in this particular case, we really changed the behavior of that overall collection of microorganisms. And more importantly, it's worthwhile knowing that in this and this batch, we basically added more of the oligoelectrolyte, but in all of these we didn't. So that oligoelectrolyte, and we know this from other experiments, they go in, they sit in the membrane, and they don't come out. If the microbe reproduces, it's carried along through a process of dilution, and the daughter cells also contain that labeling. So that's basically the story. There's a lot of mechanistic work that I've skipped, but the interesting thing is that not only are we affecting, or we can affect the way that we can extract electrons. We've also shown that the rate of electron injection is also increased, but we can also tailor the selectivity of an undefined microbial population. And this is basically born out of the fact that the molecule, its particular shape, charge distribution is complementary to that of a membrane. Once it sets into a microbe, the way that we can communicate to external electrodes is considerably modified. And these are the folks that did the work. Alex, Chelsea, are students in the group. Nate is here. I think he's going to present the poster with more details. Xiaofan did a lot of the synthesis. Heng Jin is a current postdoc. Logan is now NREL. Jenny at a startup that was begun by a student in our group. And Luigi is at Penn State. And with ICB for funding, the Gates Foundation also for funding. Thank you for the invitation, and thank you for your attention.
Thank you, Guy. We'll have a short, sorry, there's a question before we break.
Yeah, I was just interested in knowing how this process compares with I'm sure you're familiar with the anaerobic biodigestion that's used to generate biomethane, for example, from agricultural waste and other organic waste. What's the potential for this to compete with that process?
You know, I haven't seen reports that show the competition between these two. But Cambrian Technologies, they're actually installing, it's kind of like a truck-sized module. It might be that it could be easier to centrally or to distribute throughout different farms, different communities. The amount to which you can generate methane relative to what's going half a mile down the road. At the municipal plant, I haven't figured out the economics of that yet.
What about the sulfur compound?
The what?
The sulfur compound. Like wastewater, a lot of sulfur compound, H2S.
So your question is to what degree they can be decomposed, or to what degree they might be affecting?
Well, there's effect on the metallurgy, whatever.
Well, in these particular systems, in the 3 studies that we've done with wastewater, we did not do any particular pre-purification steps. So we do not think that they inhibit the function that we want insofar as the microbe is concerned. Actually, a lot of these microbes have evolved to actually deal with sulfur as an electron acceptor. So, but to the degree to which it might be fouling the electrodes, too soon to tell.
Yeah, I couldn't help noticing your current density was in milliamps per square meter.
Yeah, yeah.
Which translates into pretty small current densities. Current densities per square centimeter in the microdensity region. You can oxidize biomass species, mono and polysaccharides at very high current densities for hydrogen evolution to depolarize the oxygen evolution reaction. And that can occur at several, up to 100 milliamps per square centimeter at an operating voltage of about 0.6 volt. But that's at ambient temperature. What sort of catalysis are you looking at here for your oxygen electrode?
What kind of? We're not putting any catalyst on the oxygen electrode. It's just, I think it's no. I think no one's going to argue that microbial fuel cells is a good alternative energy source. The power density is just too low. It's not going to be competitive relative to a gallon of gas. And in both cases, you create CO2, right? Yeah. In terms of the current density that's generated from the wastewater or and this feedstock, the agricultural feedstock, I think the critical calculus to be done there is to what degree that process that you get a little bit of electricity and these fuels is, how does that relate to the way that we treat wastewater now? Because the way that we treat wastewater now is actually quite energy consuming. Some municipalities, you guys probably know these numbers better than me, it's 15, 20%. And anything that we can do to train microbes that are all around us as we speak on our surface, to do something for us that we think could be helpful. I think it really generates a new perspective on how we look at the ways that we do chemistry, how we treat wastewater, and so forth. So this is just the beginning of what I think is going to be a growing area of research in biofilm conditioning and training. Thank you.